Hi, this is Miss Litton, and this is a review. We are still on Big Idea 2, Enduring Understanding 2B. We finished 2A over 2 times, but we got that done. So now growth, reproduction, and homeostasis. Growth, reproduction, homeostasis. I'm going back. Living things are what? Organized, made of cells. They acquire materials and energy. They maintain homeostasis. They respond to stimuli and reproduce. They grow and develop, and they adapt and die. So it's saying growth, reproduction, and homeostasis require that all cells create and maintain what? What do they have to have inside? Internal environments. They have to maintain their internal environments that are different from their external environments. What right should come away in your mind, you're thinking, how do I make the internal environment, how do I maintain that homeostasis in a changing external environment? Ladies, what is that thing that goes around the outside? The cell membrane. Okay, so if you get asked that type of question, you want to go, oh my gosh, it would have to be a cell membrane. So then, that's what this is about, is talking about, talking about the cell, and we talked about the cell theory that all living things are composed of cells, and cells are the structural and functional units. And 3 cells come only from pre-existing cells. What are the size of cells? Small. Yeah. And why do they need to be small?
Surface area?
Yeah. You want to have a large surface area per volume. And on your equation sheet, you have a bunch of examples of how you can calculate surface area to volume. Okay, so you want to take a look at that. Just plug in the numbers and you'll be fine when you have to calculate one of those. Just use the right, if it says sphere or a square, whatever it says, a rectangle, calculate, plug in the right ones. Yes?
Oh, so it's kind of a general question. If there's a free day for bio before the AP exam, could we do a worksheet on a bunch?
Yeah, that's exactly what I have planned for tomorrow.
Oh, okay.
Yeah. Okay, perfect. Then we want to look at that barrier of the cell, right? And we want to think about what is the cell membrane made out of, and that's phospholipids. And what do we have inside the phospholipids? Proteins, right? And then outside we can have these carbohydrate side chains that are either attached to a lipid or a protein. What's the beauty of a phospholipid bilayer? Yes, what does that mean? Empathetic.
You have a hydrophilic head and hydrophobic tails on the inward, so different things, like if you're small and not charged, you're able to
come across, but you have to be very small and not charged. If you're large or charged, you cannot cross that cell membrane. Yes?
What is the function of the carbohydrate side chain?
Okay, that would have to do with cell recognition and also sometimes connectivity, right? And remember we talked about the MHC, major histocompatibility, that's all part of that too. Okay? And then we can see to the interior, I don't actually see much of it, but I mean there is a little bit here, that cytoskeleton that helps support the cell membrane. And then all the functions, proteins serve a lot of different functions. We'll review all of those. Yes?
I know on one of the note pages when we talked about the cell, it was like all of the different filaments and all of that. And
really detailed.
Yeah,
you won't need to know all that. Remember on that one when I showed it to you, it was just to kind of show you all the differences, but
yeah.
I said you don't have to memorize that.
I think it was like a couple test questions on your test, so I didn't know if it was something.
Yeah, no. Okay. Also, we need to point out, what's this little guy right here? Cholesterol. Who has cholesterol in their cell membranes? Animals, not plants, right? And this helps because it helps keep the cell together under warmer conditions and prevents it from freezing in colder conditions. Yeah. Okay, and so then, just like you said, if you're large or charged, you can't cross. Okay, but small molecules that are not charged can, even small polar molecules. But even then, a small polar molecule that we have is water. It doesn't. It crosses at a higher rate than it would just going through the phospholipid bilayer, and that's when we learned that there are what? Aquaporins that help facilitate the transfer of water. Is the transfer of water ever an active transport? No. You can, however, create a hypertonic environment on one side of a membrane. That could cost you energy to create this hypertonic environment, then water is going to go from a higher water potential to a lower water potential. Yes.
This was a question on one of the practice SAT
Mhm.
things that we're doing. It said glucose could go through the phospholipid bilayer. Why is that? Because isn't there a charge?
No, I don't think they meant through. I think they meant through using a protein. It usually goes as a symport, like with a photon gradient or a
Okay.
hydrogen ion gradient. Not
Okay.
photon, I don't know, light. Hydrogen ion proton, that's what I want to say.
Okay.
Like, where did I cut photon? Proton gradient, it cannot go between the phospholipids.
Okay, because I
Yeah.
was like reading that and I was like,
No, when
that
it's
can't
saying
be
through,
right.
it's saying via going through the membrane through, a protein.
Okay.
Yeah. All right, so, okay, charged molecules, okay, we hit all of that. So then we just look at the difference between passive and active. So active is going to use ATP, and that's usually against the gradient. So you're concentrating it to one side or the other. What's the difference between diffusion and facilitated diffusion? Uses a protein. Good. Is it active if it's facilitated? No, it's not active transport. It's just using your protein as a vehicle, right, to come across. Usually if it's a charged molecule, that would occur. All right, so we see simple diffusion going through the phospholipid bilayer. Then we talked about carriers. And then channel mediated, we talked about channels and carriers, right? We know our whole thing, cell recognition, enzymatic. We talked about cell junctions, all of that. And then osmosis is usually the diffusion of water across a semi-permeable membrane. Again, just showing you different ways things can cross. It's not on here, and it's probably coming up later. I just can't remember because I made this a while ago. A ligand-gated channel is when something binds that opens up a channel. Normally it's closed. We saw that with neurons, right? So the neurotransmitter would cross and bind, and then it would open up a channel, and it allows sodium to come in to allow for depolarization if it was an excitatory postsynaptic potential. Yes?
Oh, for the exam, for the systems, I was going through all the chapters.
Yes.
Again. Our main focus should be on the nervous, the endocrine, and the immune system.
Nervous, endocrine, immune, and one more. No, nervous, endocrine, and immune. That's
Okay,
it.
so
Yeah,
we-
sorry. I don't know why.
so
I
the
always
other-
want to throw in circulatory, but no. Yes.
The other
Like,
systems,
what?
do we really?
They will not, you can use them as support for, let's say, if you're doing an essay in order to make a point, let's say about homeostasis. You could bring up hormones, that'll get endocrine. You could bring up
Like kidneys and water.
kidney and water. Yeah, that would be a good
That's still-
example.
Like endocrine almost, right?
No, I mean, there's an endocrine. No, it's okay. Do you need me?
I just had a quick question.
No problem. I am recording you.
For the biomimicry hyperdoc we had last year,
Yes.
there was a 20-minute video we showed.
Yes.
Was it attached in within that hyperdoc, or was that separate? I'm trying to find it and I can't.
Oh, it was in my notebook presentation. I think I had it embedded, a link to it.
I'll go look for that.
Yeah,
Thank
so
you.
the notebook presentation that I used last year.
Okay.
And if you don't have it, let me know and I'll send it to you. I'll
Okay,
put it,
thank
it
you.
should be in our shared folder.
Okay, perfect.
Okay, so yes, talking about kidney function would be a good example of using another system, maybe to support your point about homeostasis.
Okay, but like the circulatory system, the digestive.
No, they won't ask specific questions about that, or they might give you a choice. Discuss the role of in maintaining Pick from one of these. Yeah, so you can use it to support something, but they won't ask you a direct content question on it.
Okay.
It would be a choice you could make about it. Yeah. Okay, what's going on here? I think it's follow the arrow, so it has to be endocytosis. And do you remember there was a certain kind where it's really a form of penocytosis? Receptor mediated endocytosis. So it binds to a receptor, so several receptors are attached to a molecule, and then it does endocytosis. And then the opposite of that would be exocytosis. Endo and exocytosis are always going to be examples of what kind of transport? Bulk, I need another word, active, that's what I was looking for. They're always going to cost you ATP because you are moving cytoskeleton, right, in order to make that occur. Okay then. All right, so then here I just kind of went through the examples and we talked about these. I think you're familiar with these. Channels, carrier channels, sodium potassium pump. Pumps always require ATP. Good. Cell recognition, we know about this from our immune system. Receptor proteins, this could have a lot to do, like you could tie this into the endocrine system, right? Which one, what type of hormones require receptors on the surface of the cell? Protein hormones, right? They stand at the door and knock and bind, and they trigger a secondary message inside. It could be a metabolic reaction that's occurring inside, or it could trigger a gate to come open so that calcium floods in. There are different ways that it could trigger having a secondary, what do I want to say? Messenger, thank you.
So when a neurotransmitter goes across the synaptic cleft and binds to a receptor, so the neurotransmitter is a protein, and that's a signal transduction pathway.
Yes.
Okay.
You got it. It's examples of all of those things.
Yeah.
Okay, enzymatic. You're a good one for there with your hormones. Do you remember how we talked about cyclic AMP? Do you remember how we talked about how that triggers, we talked about adenylate cyclase and how that triggers a secondary message and increases glucose availability to the cell. And that would be, again, that second messenger example. Okay, and then aquaporins, allowing water to move at a faster rate than what you would expect at a cell membrane if it was just going across through the phospholipid bilayer. We talked about peripheral proteins being involved because they don't cross the membrane, so they're not used as channels or carriers, but they could just be used at the surface, and it could play more of a structural role. Okay, be your favorite thing you're looking forward to doing this summer. It's everybody's always. Just a second, I will remove. I'm going to pause you for a moment. Yeah, I'm sure you can find one.
Or in the back of the books.
Yes. Okay, so carrier proteins are specific. You want to remember that. Specific in their action. Okay. All right, so the next thing we need to talk about is osmosis. This is critical, right? Water. The diffusion of water across a semi-permeable membrane. And remember, you're always going from a higher water potential to a, unless what? What kind of situation do you have no net change in water? Isotonic, perfect. Okay, so we looked at a cell in an isotonic solution, you're not going to gain no net change overall. But if you are put in a hypotonic solution, then water is going to go from the hypotonic solution to the interior of the cell, which is hypertonic. Right, we're going to have that movement, and that's great for a plant cell because the water is going to move into the vacuole. The vacuole is going to enlarge and push against the cell wall, creating turgor pressure, which is good for the plant because the leaves will be what? Upright. So it's not ever going to be bad for a plant, right? Because they have a cell wall that exerts turgor pressure and pushes back. Having plenty of water, the only time you might see outcomes of that is like eutation, when it gets water, so much water it gets pushed out the ends of the leaves, right? But it's still not damaging the plant. It's still upright, fully able to do photosynthesis. It's not worried about water loss due to transpiration. For an animal cell, that can be toxic, right? Because it can kill the cell because we don't have anything to protect it. We can't withstand changes as well as maybe a plant cell could. Okay? In a hypertonic solution, it crenates, and you can see it shrinking and shriveling. The vacuole has become so small it's going to pull away. There's nothing to push on the walls, no turgor pressure that's positive. It would actually be almost negative turgor pressure, right? And that's going to cause the plant to wilt. Yeah. All right, so then talking about the equation for water potential. It's a factor when you want to measure water potential of two things, pressure potential and the solute potential. Meaning, how many osmotically active substances do you have? Remember, pure distilled water is 0. Pure distilled water is 0. You add just one salt molecule and it's going to go what? Negative, yeah. It's going to go negative. Okay, and so you're adding both of those together to give you your overall water potential. And we learned how to calculate the solute potential, right? We have the equation for that. So we can do that based on the molarity, right? The temperature. whether or not it ionizes or not, and then we have a constant, what is it, 0.0831 liters per bars per moles per Kelvin. I don't know that I have it exactly right. But anyway, you multiply those together, right? And then that is the solute potential. And we know if the plant is neither gain nor losing water, right? If it's not, it doesn't mean it's isotonic, right? Because the cell wall can push back. But we know that the pressure potential would be equal to 0, so therefore your water potential is equal to the solute potential. And that's what we solve for. I don't know if I included it in here, but I'm thinking about it. That's what we solved for when we graphed, right? We had our graph and we said this was 0, right? And this was a positive change, and this was a negative change. And so when we drew our line over here, we marked molarities of the solution that it's sitting in. And so one thing you can do, I read about, like, you know when you have to read graphs on the AP exam and you don't have a ruler, use your pencil. You know how pencils have the flat edges to it? Bring a pencil that's like a real pencil. Yeah, these kind of pencils. You see how it has a flat edge to it? And then use that. Use the point, and you can see if your pencil's straight or not, and use a pen and draw a line when you're reading their graphs. Right? Do you understand what I'm saying? So you would put that flat edge on there, and you'd go, okay, that looks like it's right about there. Then draw your line so you can see where it crosses. Okay? And then once we had that value right here on the molarity, whatever it was, maybe it was 0.23 molar, I don't know, it depends on it, right? Then you could multiply everything else together to calculate out, right? If it's something like sugar, it's 1 is the ionizing, right? Salt would be 2, right? Okay. And here you go. And I'll do a little pause, give you some time. Okay, so let's take a look at this. Water is always the solvent, and whatever you're dissolving into it is the solute. A hypotonic solution contains the most solutes, okay? So keep in mind, okay, if I made a solution, I'm going to put a red blood cell in here, and if I say it is a hypotonic solution, if this is hypo, then this has to be what? Hyper. And the question says the hypotonic solution contains the most solutes. It contains the least. Okay? Because if it's hypotonic, it means the other side of the membrane has to be hypertonic. You could correct this by saying contains the most what? Solvent. Right?
So it contains the most.
Yeah. And then here, when a cell is placed in a hypertonic solution, so now we're going to take this cell that we just put in here, okay, and we're going to put it in a hypertonic solution, so it has more stuff out here. So it is hyper, and then the cell is hypo, right? Water must flow from the hypo. So this cell should shrink. So when a cell is placed in a hypertonic solution, the solute exits the cell. Okay, remember solute and solvent. So solute is the stuff that's dissolved. Is the concentration gradient wanting to make the solutes go from inside the cell to out? No, they're going to go from a higher concentration. If they can move, these green things would come in here, right? They're going to go from a higher concentration to a lower concentration. So the solute exits the cell? No. To equalize the concentration on both sides of the plasma membrane. How could we change this to make this correct? The solvent would exit the cell because it's going to go from a hypotonic to a hypertonic. So water exits the cell towards an area of a higher solute concentration. Ouch. Okay, good. All right, next, let's see. Oh, we're almost done with this big idea, or enduring understanding. Facilitated transport, we already talked about this, going from a higher concentration to a lower concentration using a protein. Active transport, the one you're going to want to use is the sodium-potassium pump, and I would talk about a neuron. And then we did endocytosis and exocytosis. We already reviewed that. Follow your arrows. And then this is receptor-mediated endocytosis. And I have a question there, but oh, wait. Oh, I thought I was close. Am I not? Oh, no, I am close. Okay, yeah, kind of, we are. So then this is talking about the endo-symbiotic hypothesis, where one soul, this sweet soul right here, one cell engulfs what? Another cell, right? And then those became the organelles, and our evidence for that is they have their own, mitochondria and chloroplasts have their own DNA, right? And they are and they do their own protein synthesis, and the ribosomes are the same size and shape, and the chromosome is circular, just like bacterial cells.
Don't they also have a double membrane?
Yes, and double membrane. Good. Okay, and then just knowing the parts of the animal cell and the plant cell, and again, structure and function, right? Structure and function. A plant cell would have a larger vacuole because that vacuole is important to maintain the walls being upright so it can create turgor pressure for photosynthesis. They both have mitochondria because they both have to do cellular respiration. You only see the chloroplast in the plant because it does photosynthesis. We don't know about centrioles, churros. It may be related to cilium flagella, who knows? But animal cells have that. Love you so much. They both have a nucleus. They both have ER because they have similar. You're going to want to be able to, if you had a question like that, you're going to say they have similar metabolic needs. They both need to make proteins, so they both have ribosomes, right? They both need to process, package, and transport. That's why, see what I'm saying? So look for similarities and why you would have that particular structure. Yes?
Should we know, like, every single part of the plant cell and animal cell?
I would be familiar with like key organs and how they help maintain homeostasis. You would know about the nucleus because that's where the DNA is protected, right? You would know about ribosomes because they facilitate protein synthesis, right?
What about like lysosomes or peroxisomes?
Okay, so you would want to know about lysosomes because that helps with the digestion, but that's not like a system you need to know, but you need to understand that you can break down molecules. And again, the pattern there is that reaction is kept separate. It's compartmentalized within the cell, and how compartmentalization can lead to specialization, right? Those are the kind of things you're looking for. What is the significance of that, having that as a separate process?
Is compartmentalization, is that part of Beta-A3?
I don't remember, because you can kind of pull it to several different ones, because you could talk about big idea 1 and the evolution of compartmentalization and the evolution of the eukaryotic cell. I don't think you could talk about it, I guess you could talk about it in systems and how you build. So, I mean, you can just look for connections between that. Okay, so we know about the nucleus, we know about the structure and organization of DNA, we know about ER, and keep in mind that if all you did was endocytosis, you would deplete your cell membrane, right? So by doing exocytosis, you're adding back to the cell membrane. And you're going to get that a lot of times from the smooth ER, or any vesicles that went from the smooth ER to the Golgi, right? Okay. And this is just showing you ribosomes and the connection between lysosomes and an endocytotic vesicle, and how nutrients are digested, and then how you can do exocytosis to get rid of anything. That's it. And now we are on that one. Okay, good job. Yay! Have a piece of toast. Hope you're reviewing.